Welcome to my poster presentation titled Quantitative Analysis of In Vitro Micronucleus Concentration Response Data for Genotoxicity Evaluation and Chemical Prioritization. The in vitro micronucleus assay has been recognized by the OECD for assessing genotoxic carcinogens. This assay detects DNA fragments or an extra chromosome in cells after cell division, known as clastogenic and antigenic mechanisms, respectively. The presence of these visible micronuclei in cytoplasm is an indication of chromosome damage. The US EPA has produced micronucleus datasets using a high-throughput approach. Very briefly, Chinese hamster ovary cells were treated with 292 chemicals on 96 well plates. Each chemical was tested with 20 concentrations, ranging from 0 to 200 micromolars, plus and minus the rat liver S9, and the results were reported as percentages of micronuclei or hypodiploid counts. In this project, we proposed a decision tree-based informatics approach to evaluate each chemical's micronucleus and hypodiploidy frequencies and classify them as positive, negative, and inconclusive. An in vitro-to-in-vivo extrapolation or IVIVE approach follows to assess the chemical's potential risk. In our decision tree-based approach, to be called positive for micronucleus, a chemical needs to satisfy the following conditions. First, micronucleus responses of two consecutive concentrations have to be greater than the 95th percentile of batch-specific solvent controls. Second, the observed increase in micronucleus frequency needs to occur at non-cytotoxic or adequate cytotoxicity levels. Third, having a dose-response trend with a non-parametric test. Finally, having a benchmark dose lower than the highest concentration tested. An inconclusive decision is made when the first two conditions are false. The rests are considered negative. By applying the decision tree-based approach, we identified 157 unique chemicals with positive micronucleus responses, 25 negative, and 110 inconclusive. We applied the same decision tree-based approach to the hypodiploidy datasets. 209 chemicals were identified as positive, 27 negative, and 56 inconclusive. In 2011, Bryce et al. Proposed an approach to assess hypodiploidy datasets. In their approach, a positive call is based on if micronucleus fold change was greater than 3 and hypodiploidy fold change was greater than 10. We manually examined the 15 positive chemicals based on the Bryce approach and verified that 6 were true antigens and 2 were potential antigens based on published results. We performed IVIVE analysis to convert the BMDs to oral administered equivalent doses using the HTTK package in R. AEDs were compared to exposure estimates and traditional genotox PODs. This histogram shows that the majority of the positive clastogens have an AED value lower than the traditional genotox POD. We can prioritize chemicals for further evaluations by computing the bioactivity exposure ratio, or the BER. A lower BER is considered as having a greater potential concern for risk assessment. In these plots, we see that the AEDs provide a conservative POD relative to traditional in vivo genotox endpoints, with a few exceptions. In conclusion, we proposed a workflow to assess high-throughput micronucleus datasets. This workflow starts by classifying the chemicals as positive, negative, and inconclusive for micronucleus, followed by IVIVE analysis to assess their potential risks. To the best of our knowledge, this approach has not been applied to micronucleus datasets. Our proposed informatics approach provides a useful analytical workflow to risk assessment applications.